The overall goal of this procedure is to grow in vitro modular networks consisting of spatially confined functional interconnected neuronal circuits. This is accomplished by first preparing the PDMS stencils to pattern a protein layer to promote cellular adhesion over the culturing substrate. The second step is to clean the culturing substrates, specifically the surfaces of petri dishes, cover slips, and multi-electrode arrays. Next, the PDMS stencils are deposited on the culturing substrate, and the desired adhesive protein layer patterns are created. The final step is the plating of the neuronal glial cells. Ultimately, multi-electrode array recordings and calcium imaging are used to show the dynamics of the achieved modular neuronal networks.
This method can help answer key open questions in the neuroscience field, such as investigating the dynamics of communication among neuronal assemblies under well-controlled experimental conditions.
Polydimethylsiloxane, or PDMS, is made by first mixing 1 part curing agent with 10 parts of base agent. After 5 minutes of mixing, move the mixture to a vacuum chamber for 15 minutes. After 15 minutes, check for bubbles and return the solution to the chamber for 15 more minutes. Next, prepare a wafer on the spin coater. Open the gas knobs for nitrogen gas, put a wafer on the spinner, and secure it in place with the vacuum.